One of the main goals of SILICOFCM project is to establish the connection between genetic analysis and heart functions through a sequence of experimental and computational elements. The influence of a certain genetic mutation on the protein structures and interactions is investigated through a number of experiments and very complex computer simulations. Bix2Mus workflow represents a linkage between genetic analysis and music-OSL, musico, and MP surrogate results presented in the experimentation database. Once the genetic analysis of a new patient is conducted, the user provides a VCF file and runs the workflow. In the first stage, the workflow runs the annotation pipeline in order to detect possible genetic mutations. During the analysis, the user is informed about progress and status of the pipeline execution. When the execution is finished, the user can download results of the analysis or to filter virtual experimentation database in order to find the experiments with the detected mutation. The knowledge collected from both real and in silico experiments conducted within SILICOFCM project are stored into the virtual experimentation database, which contains a number of virtual experiments for various types of protein mutations, performed using music-OSL, musico, and MP surrogate modules. Filtered database will show all the experiments conducted on the muscles with the selected mutation, so the user can explore the influence of the mutation on the simulation results. Once the required experiments are shown, using the buttons Inputs and Outputs at the left-hand sides of the table, the user can download the input parameters and the results of the corresponding simulations performed by one of the SILICOFCM modules. Using the downloaded data, the user can perform simulations in the corresponding SILICOFCM module with the same parameters and test how variations of certain parameters influence sarcomere response to an excitation. Input parameters of MP surrogate model can be further used as an input to ALIA or PAC, finite element solvers, as parameters of the micromodel. This way, the user is able to test the influence of a detected genetic mutation on the function of the whole heart.